All right, welcome everybody on this special occasion of the seminar, and I have the great pleasure to introduce our seminar speaker today, who is Anett Kiss, who's, as you can guess from her name, she's a Hungarian-born scientist who is going to talk about her PhD work done in Oxford. Anett, actually, as I mentioned, was born in Hungary, but later on she moved to Italy during her high school years, and from there she moved to Edinburgh to do her undergraduate studies, and then from there she moved to Oxford to do her PhD studies under the supervision of Ben Sheldon around the famous woods of Oxford, where many excellent ornithologists, including David Lack, did his studies, and this is the place where Anett worked, and she's going to talk about her research. So thank you so much for accepting our invitation, and the floor is yours.
Thank you very much for that introduction, and also thank you for inviting me to talk about my PhD work, where I investigated life history trade-offs and the role of feather quality. So just to give you a bit of an introduction, I think we can all agree that there is an amazing amount of variation in environments all around us. And all of these require a wide array of unique solutions from organisms in order to cope with the many challenges that they pose. And life history theory provides us a framework to understand how optimal solutions related to the survival, growth, and reproduction of organisms are shaped by both extrinsic factors as well as intrinsic constraints. So as an example, there are very different optimal solutions that organisms adapt in terms of, for instance, their lifespan. So adult mayflies only live for about a day, whereas tortoises can live longer than 100 years often. We also see very different optimal solutions related to growth, such as the rate at which organisms mature. And we also see very different solutions in terms of reproduction, such as some species reproducing multiple times in their lifetime but at lower effort, versus other species just reproducing once but at very big effort. And all of these life history traits are influenced by extrinsic factors. For instance, food availability can affect development rates, for instance in amphibians. And they're also underlined by intrinsic constraints, such as allocating energy between competing behaviors, such that you can't both grow and reproduce simultaneously. But we also see differences in life history traits, even between individuals of the same species. And seasonal habitats in particular require the periodic fine-tuning of different behaviors of these organisms living in seasonal habitats, often because they rely on some sort of ephemeral resource to complete different stages of their annual cycle, such as seasonal reproduction. And a very classic example, and one which I'm very familiar with, is how fitness in great tit is maximized when the timing of the nestling provisioning coincides with the timing of the winter moth caterpillar availability, which is only available for a short time each spring. But this optimal timing of raising chicks is actually preceded by several other life history traits, such as decisions on when to start their reproduction in a given year, how many eggs to lay, as well as when to start incubating and for how long. And because of this ephemeral nature of the resource, this creates an energy and time limitation for the individuals, which means that now they have to balance trade-offs between these competing behaviors. And therefore, the optimal timing of chick rearing will depend on a careful balance between the trade-offs between these different life history traits. So this leads to the first part of what I looked at in my thesis, trying to understand how individuals optimize multiple life history traits simultaneously. But especially for species that breed multiple times in their lifetime, year on year, they are not only faced with short-term trade-offs, such as between the number and quality of their offspring, but they also need to consider longer-term trade-offs. For instance, dividing energy for their current reproduction as well as their future reproduction and survival. So this leads to the second part of what I looked at in my thesis, trying to understand what mechanisms might mediate both the short and the long-term trade-offs between life history traits. And a large body of research has suggested that it's some form of individual condition that's linking these life history traits together. And a lot of research has looked into immune function related traits like oxidative stress or response to infections. But as these traits are often quite complex to measure or underpinned by complicated pathways, some researchers have also suggested that instead we could look at some morphological trait that's periodically fixed in time. So for instance, the leaves of deciduous trees, or the fur of animals, or the feathers of birds. Because birds annually regrow and mold their feathers each year. And so for the duration of the time when they have their feathers, it's fixed in that quality, and it can potentially affect many different life history traits along the way. So as my study species are birds, I, of course, focused on variation in feather structural quality. And for feather structural quality to link different life history traits, it needs to respond to energetic and time limitations. And we do see some evidence that, for instance, a low-quality diet can lead to different types of feathers with different structures, for instance, lighter and shorter feathers in house sparrows. And we also see some evidence that time limitations can also affect the quality of feathers. For instance, starlings manipulated to mold their feathers in a shorter amount of time resulted in growing feathers that were lighter and shorter as well. But also for feather quality to link life history traits, both over short and long term, we also need it to impact on functions that are important for survival. For instance, insulation. And we see some evidence that northern populations of great tits have denser and shorter feathers, potentially as a result of providing better insulation in the colder winters. So, to remind you all of the two main aims of my thesis, I looked at how individuals optimize multiple life history traits simultaneously, and then looked at the mechanisms that might underlie both short and long-term trade-offs. So, to address the first question, I looked at how life history traits are optimized in response to temperature variation in seasonal environments. And for this, I used an agent-based model in R, where I simulated the daily behavioral decisions of a population of great tits. And although it was very much parameterized for great tits, it can easily be adjusted for other species that rely on some ephemeral resource for reproduction. So these are the different behaviors that I simulated, for instance, relating to foraging intensity, or the timing of egg laying, the clutch size, the incubation start and duration, as well as the intensity of caring for offspring. And birds also had the option of abandoning their reproductive event altogether. And I then defined an optimal strategy as a sequence of behaviors that maximized both offspring number and quality. And of all the behaviors, I focused on what's the optimal combination of laying date and clutch size that maximize fitness under a given environment. And I focused on these two key traits because they are tightly linked and influence the timing of chick provisioning in great tits. Because great tits lay one egg each day, and that means that a larger clutch of eggs needs to be started earlier on in order to reach the same hatch date. And as we discussed, it's very important that they time their reproduction correctly during the period when caterpillars are available. So in terms of looking at what are the optimal solutions under different environmental contexts, as an environment, I defined a combination of temperature, annual mean, and seasonality, where seasonality is just the difference between the annual maximum and minimum in temperature. And then this temperature cues the availability of caterpillars for providing for the chicks. But the temperature also cued some of the reproductive behaviors, such as the egg laying or the incubation. And you can see this is a range of different environments that I tested, so a range of different combinations of annual seasonality on the y-axis and annual mean on the x-axis, but you could define any number of combinations of them. And what you can see from here is that both as you move from bottom to top, as seasonality increases, but also as you move from left to right, as annual mean temperature increases, so the blue line, you also see that the caterpillar peak becomes shorter. So these are just some general trends of how these two variables are related. And this is how I'm going to represent the results of what the birds are doing and what are the optimal solutions that emerge under the different environments. So here, an optimal solution is a combination of laying date and clutch size that maximize fitness in that environment. And each dot represents the emergent optimal strategy in a given simulation run. And here I'm showing you 100 simulation runs. And the vertical and horizontal lines represent the mean across all of these optimal solutions. So to break it down into more manageable chunks, we can first look at how the optimal solutions vary along temperature seasonality. And what you can see that I think is quite evident is that there is variation in the range of optimal strategies that emerge. So you can see that as you move from bottom to top, as seasonality increases, the range of possible strategies become limited, both in terms of clutch size and lay dates. So you can see that only lower clutch sizes emerge as optimal under higher seasonality, as well as the range of lay dates that are possible that lead to successful reproduction are also shortened. And just to remind us of what the environment underlying this looks like and why these patterns might emerge, we can see that an increase in seasonality increases the rate of spring warming, and that causes a sharp and short peak in caterpillars. So as you move from bottom to top, you see it becoming shorter as well as peakier. And now looking at what happens along different values of annual mean. So here the seasonality is the same, but we vary the annual mean temperatures. Here you can see that there is still some variation in the range of optimal solutions that emerge. But annual mean seems to limit only the range of lay dates that emerge. You still see a range of different clutch sizes being optimal under those environmental conditions. But lay dates become much more restricted. And so if we look at what happens in the environment, we see that a higher annual mean just means an earlier spring in terms of advancing the emergence of caterpillars, but the window of caterpillar availability remains relatively similar. So this is just kind of looking at these trends that we've already discussed in a slightly different way. So on the left, I'm showing how the optimal clutch size varies with annual mean temperature on the x-axis, and the different colors represent different annual seasonalities. So yellow is the lowest and pink is the highest seasonality, and the same for laying dates. So you can see that laying date is driven by both mean and seasonality and temperatures, but clutch sizes are only really driven by seasonality in our models, because the lines along the annual mean temperature range are all flat. And specifically, we can see that an increase in annual mean temperatures shifts optimal laying to be earlier, but that this shift towards an earlier optimal laying happens to a lesser degree under higher seasonality, so moving from yellow to pink. Whereas we see that with clutch size, high seasonality, so the pink in temperatures, selects for a lower optimal clutch size than under lower seasonality. However, this seems to be in contrast to what we observe in the wild. And here I'm showing comparison between our simulated results, so those lines are shown in pink, and also the observed trends. And these trends come from the long-term MITEM data set. So this includes over 60 years of data on the reproductive behavior of greytits, from which I managed to calculate these lines of what happens to their laying time as you move along different values of annual mean temperature and seasonality. And you can see that laying dates correlate well with the simulated results, where observed trends also show an earlier laying in response to higher annual means, but this advance happens to a lesser degree under high seasonality. But as you can see, the clutch size trends are quite different, and in fact, under high seasonality, the observed birds tend to lay larger clutch sizes. And yeah, I'm happy to discuss reasons why I think this might be at the end of the talk if anyone's interested. So, to summarize, different aspects of the environment may drive independently different life history traits towards different values, but these simulations showed that ultimately fitness is maximized under an optimal compromise between multiple traits. And we showed that the range of possible strategies really depends on the environmental context. So now moving on to the second part of looking at what mechanisms might mediate both short and long-term trade-offs in terms of variation in feather quality. I first had to develop a method of measuring feather quality. And for this, I developed a field method and wrote an automated R package for collecting and analyzing feather quality metrics. So for this, I used a digital microscope that you can bring around in the field and you just plug into a laptop, so very portable. And you can then take images of the bird's feathers in vivo, so no need for plucking. And you can then later analyze these images for commonly used feather quality metrics, such as the width of the rachis, or the width and density of the barbs, or the density of the barbules. And so this provided me with a non-invasive and time-efficient measure of feather quality, because as we were ringing the birds, I could just put them under the camera, take pictures, one, and it was done in no time. But then I had a backlog of images, and so I needed some way of analyzing these efficiently. So I wrote an R package that I will want to later release as a package for anyone to use, which automatically counts these different structures in the feathers. So it includes different functions like obviously preparing the images for the quantification, like changing it to grayscale, increasing contrast and things like that. And then the first step is to identify the rachis in the image to orient ourselves of where the feather is. Once we have that, we can also measure the width of that rachis. And then we can pre-specify at what distance from the rachis we want to measure the other feather quality metrics to make it standardized. So along that crop line, we can then count the number of barbs to estimate barb density, as well as the width of the barbs. And once we have the position of the barbs, we can move on to counting the barbules along them. And most of my images were taken on little birds that you can just hold in the hand, but I also took some images of swans. You need a bigger team for that, holding down the bird, but it works just fine. But in those images, because they have such big feathers, actually when you take the image, the rachis is invisible, because as you zoom in, it would be all that you can see in the image. So if you're interested in the density of barbs and barbules, then you won't have a rachis in the image. So I'm also working on functions that can detect the orientation of the feather in the image so that we can do the next steps, like defining where to measure and then identifying barbs and barbules. So they're very tiny. I don't think it's very visible, but on the left are some examples of when the automation works nicely. So you can see that it correctly identifies the whole width of the rachis. And you can't see, but those are examples of it identifies each barb correctly and also draws the correct lines through the barbules. But there are some common errors that can hinder the analysis. For instance, if there is glare in the image or uneven lighting, or if there's any like damage or dust in the feathers, it's best to avoid and take pictures somewhere else. And I suggest some ways of trying to deal with this both before you take the images and also after analysis. So now actually asking how feather quality might mediate both short and long-term trade-offs. First focusing on the short-term trade-offs. I looked at how plumage structure might act as a mediator of the trade-offs between offspring number and quality, using an experimental study of blue tits. So for this, I carried out a brood size manipulation experiment and then followed the individual growth trajectories of hundreds of blue tit nestlings. So brood size manipulation occurred when the chicks were 2 days old, and I moved them around in these insulated carrier bags where I custom built them a little nest with moss and wool and put in some hand warmers to keep them warm. And then I swapped chicks between pairs of nests in such a way that one pair of the nests increased in brood size by 2 chicks and the other one decreased by 2. And I then went back and followed their growth in terms of their weight, their wing length, as well as measuring their barbule density once their feathers were out. So once again, I used my digital microscope approach to take images, and then here I'm focusing on the results with barbule density. And to test whether there was an effect of the manipulation on the different morphological metrics, I used mixed modeling approach in R, where I fitted as a response the different morphometrics. So I'm going to be focusing on weight. All of these metrics are taken on day 15, which we take as like the final measurement because the growth of the birds tends to plateau at this point. So I fitted weight, wing length, and barbule density separately. And as fixed effects, I included the manipulation as a continuous variable in terms of the actual number of chicks that the brood size was manipulated by, as well as including clutch size as a measure of the original parental investment, hatch date, because we see every year that the later it gets in the season, the worse quality the chicks become, so it has a significant effect on their well-being. And I also included year because I measured these morphometrics and did this experiment 2 years in a row. And as random terms, I included nest of origin and nest of rearing. But I was also interested in the clutch size specific effect of the manipulation, because you can imagine that adding 2 chicks to a nest of 5 chicks is a different sort of manipulation than adding 2 chicks to a nest of already 13. So I also included a multiplicative effect between manipulation and clutch size. So what you can see in each of the results, I will be showing on the left the z-scored value of the change, and on the right the raw results. And on the x-axis you can see the number of chicks that the brood was manipulated by. So you can see that adding chicks to the nest overall decreased their weight, but that when you look at this in more detail, you can see that this trend is mainly driven by what happens at lower clutch sizes. So here I'm showing each panel is a clutch size, so in the middle is what happens at the mean clutch size, and then what happens at 2 standard deviations either side of it. So you can see at lower clutch sizes, and mid clutch sizes, that's where we see this like sharp decline in weight as a result of the manipulation, but we don't really see that at higher clutch sizes. In terms of wing length, we also see some sort of a negative relationship, although not exactly significant. But yeah, it's not a very clear pattern across all of the clutch sizes. We again see it mostly at the lower clutch sizes, low to mid clutch sizes. And then in terms of barbial density, there is some sort of a trend towards an increase in barbial density when you add chicks, but again, not significant. But if you look closely, some of these comparisons are significantly different between an increased nest and a control nest. I should have said the blue one is always the decrease, it's quite small in the image. The blue is always the decreased nest, the control where we didn't change brood size is the yellow, and then the pink is the increased nest. So you can see that this trend towards adding chicks, increasing the barbial density of those chicks, mainly happens at the larger clutch sizes. So there seems to be at least some kind of evidence of trade-offs between offspring quantity and quality, because we see it affecting some of the morphological traits measured. And we were thinking that it might mean that under increased competition, the chicks might prioritize investment into more structural morphological traits like feathers, rather than investing into transient morphological traits like body weight, and that might explain this negative relationship between a decrease in weight but an increase in barbial density. But once you control for the correlation with wing length, with both weight and barbial density, it actually removes even the slight trend that we see in barbial density, but the trend with weight remains the same. So actually what this means is that given the wing size, the barbial density just changes according to wing size, and so there might not have been any differential investment as a result of the manipulation. But I have to highlight that I'm not sure whether we can conclude anything very conclusively, because the wing length that we measure on day 15 isn't really their final wing length, as their feathers aren't fully grown at that point. And also we measure the wing length, not the feather length. So a wing length can be made up of different skeletal sizes and also different feather sizes. So I think, I'm again happy to go into more detail about this, but I think our methodology limits us from actually being able to tell whether any differential investment towards barbial density occurred. So now moving on to the long-term trade-offs. I looked at how manipulated foraging costs can alter individual morphology and thermoregulation in zebra finches, and linked feather structure to insulation efficiency. So for this, I worked with a group based in the Netherlands, where they have a captive zebra finch population that they've manipulated for a long time for foraging costs. So the birds are divided into two groups, one with benign foraging costs and the other with harsh. And benign foraging costs mean that they have access to food hanging from a ceiling, and they can just sit at perches and eat that food, whereas the harsh treatment birds need to constantly fly back and forth from the food source. Then I also measured, using my usual approach with the digital microscope, took pictures and measured the different feather quality metrics. But I also used a thermal camera to record the surface temperatures of the birds, and then use this to calculate some proxy for insulation. So what I did was contrast the temperature on the eye, which is an uncovered surface, and one that correlates with the internal body temperature, and contrast that with the temperature on the body, so on an insulated surface. And as another measure of thermoregulation, I also looked at metabolic rate, measuring the oxygen consumption of the birds in respiratory chambers. And yeah, conducted this across 2 years. So to look at the effects of the foraging costs on the morphology of the birds, I once again used mixed models and fitted the response as either weight or barbial density. These are the two morphological features I'm going to be focusing on. As fixed effects, I included the food treatment, sex, age, and also year, because I measured them across 2 years, and random term individual identity. And then for the thermoregulatory metrics, it's all the same effects, but each of them were measured in separate years, so I don't need to fit a year effect because they were all measured once each. So what we see is that foraging costs alter individual morphology. In yellow, you can see the change in weight, and it's broken down into males and females, but overall, if you just look at the difference between benign and harsh treatment, you can see that they experienced a reduction in weight. And they also experienced sort of a reduction in barbial density, again, not significantly so. And in terms of the thermoregulation, in orange you can see metabolic rate, and in pink you can see insulation efficiency. And both of these tended to decrease under the harsh treatment. So harsh treatment birds had lower metabolic rates, and they also had reduced insulation. So I should say, yeah, a larger difference between the eye and the body temperature means a better insulation. So I then wanted to ask how the different morphological features actually influence thermoregulation, because ultimately what I want to see is how feather quality relates to insulation. So as response, I fitted the thermoregulatory traits like metabolic rate and insulation, and as fixed effects, I tested different combinations of the quadratic form of weight and barbial density, as well as food treatment, sex, and age. So what you can see is that metabolic rates are only driven by weight, not barbial density. And we see this positive relationship where larger birds have higher metabolic rates, which is a pattern that you see across taxa. So it gave us some validation that we were measuring meaningful things. And then what I found very interesting was this relationship between barbial density and insulation, which seems to suggest that it's some intermediate barbial density that provides the best insulation overall. And I also wanted to see whether any of the morphometrical differences led to survival differences in the birds, but none of them did. None of the shifts in morphology led to any survival probability differences, but age did. So to summarize, the foraging costs altered weight more than barbial density, and this is similar to what we saw with the brood size manipulation, that effects were more visible on weight than barbial density. So once again, the birds might be prioritizing keeping structural morphological features at a good quality, and if faced with harsh conditions, they turn more towards varying their transient morphological features like body weight. And this might be especially true for harsh condition birds, because they need to fly much more. So they need to have good quality feathers to be able to fly efficiently. Of course, the effect on weight could also just be simply the result of resource limitation, that if you have higher costs trying to get resources, then you end up getting less of those resources. But it might also show some energetic adaptation for more efficient flight. Carrying less weight means a more efficient flight. And in terms of why this counter-relationship exists between barbial density and optimal insulation, I was trying to think of the different insulatory mechanisms and how they act. And I think they might be driving barbial density towards different values. So actually, this in-between value might be kind of balancing both of these different mechanisms, such as a higher barbial density might be better for lowering the rate of convection. So that's like air movement, moving warm air away from the body. So if you have a higher barbial density, then you create a more homogeneous surface to stop air movement. On the other hand, if you have low barbial density, then you can have more space to trap air, which is of poor conduction, so it can limit the loss of heat in that way. So to summarize it all together, how do individuals optimize multiple life history traits simultaneously? We illustrated that fitness is maximized under an optimal compromise between multiple traits, but that it depends on the environment. And in terms of whether feather quality might be a good candidate mechanism linking trade-offs over both short and long term, we found some evidence that it might be influenced by energy and time limitations. So we saw that brood size has some effect on bluted offspring feather quality, and foraging costs also have some effect on zebrafinch feather structure. And then seeing whether feather quality impacts functions essential for survival, we looked at insulation and found that barbial density is linked to insulation in zebrafinches, but the direct links between feather quality and survival still remain unclear. So I would just like to thank my supervisors, Ben, Ellen, Charlotte, and also my collaborators, John, Chris, Simone, and Marianti. And of course, my funders, NERC and Oxford, and also the whole Sheldon group and the field team. And thank you for listening.
Thank you, Anett, for this very interesting and also visually very appealing talk. I forgot to mention actually this during the introduction. I should have that Anett has some fantastic, apart from the scientific skills, some fantastic artistic skills as well. So she makes her own illustrations, and also she makes some, if I'm correct, some bird-themed handmade jewelry. So if we run out of the scientific questions, you can also ask questions.
Christmas is coming. So.
Yeah, Christmas is coming. All right, questions?
No, he's the boss. Yeah. I'm glad to see that Ben is preparing for a climate change by developing isolating buffer on his face. Yeah. So very well done. It's a very interesting PhD, a very interesting presentation. So just two quick things. Now, about the short-term life history trade, of course, the trade-offs is one of the big Oxford, Croning and Link, Simon, and they work together for a very long time. So when you actually manipulated brood size, which is of course is very clever, and add the barbial density to the measures, I was just wondering whether one interpretation is that the feather quality is actually genetically programmed, because you get smaller babies, yeah, in response to clutch size, but brood size, but the barbial density increase. This could be simply because the relative, for a smaller body, the individual produces the same sort of feather output, right? It's not necessarily trade-off. Yeah, it's not trade-off. It's actually just as one feature, baby weight, is actually traded off, responding to the clutch size, but the other isn't. So it's actually not entirely trade-off. Number two, zebrafinches is actually adapted for arid environment in Australia. So in that environment, you know, it's native environment, heat is the problem. So they have to think how to lose heat rather than how to buffer themselves against heat. So in this situation, I was just wondering whether actually poor feather quality is actually a good thing, which is consistent with the optimal graph, if I remember. Yeah, you had sort of the barbial density at mid, insulating capacity at mid density, because if it's super hot, they really want very loose feathers because they just burn, yeah, just burn, you know. So two destructive or sort of constructive ideas.
Yeah.
Well done.
I'll address the first. So, because I manipulated brood size by swapping chicks between nests, I actually also estimated the genetic and environmental components. So, if I switch to my other presentation, right?
Yeah, you
so
can. Yeah.
Some extra figures.
That's a nice bird. Yeah, very nice.
Yes, here I'm showing the proportion of variation explained by genetic components in yellow and environmental components, so nest stock varying in orange across the different metrics. And so here you can see in bardial density that overall, from these calculations, it seems that the environmental component is bigger than the genetic. And even when you break it down into even smaller components, because I took multiple measures per individual, so I could also estimate between and within individual variability. Even once you account for those, it removes a lot of the genetic and environmental effects, but still the environmental is bigger. So I would argue that yes, it is influenced by genetic effects, but actually according to these, the percentages are rather small. So.
Can I just comment one of the suggestions of Bill Sutherland? Yeah, he's in Cambridge, so he's in an opposite castle. Bill checks out every PhD which has a pie chart without number of individuals, right?
I see.
Oh, sorry, no, it's unrelated.
It's 360.
I know, you're a massive sample size. Yeah, very well done.
Oh yeah, actually that's a good point. I should put that.
No, don't worry about it, yeah.
But yes.
The other one is zebrafinch, so it's basically two-type barb with two-type feathers may not be suitable, especially for a desert-living creature, which have a problem, especially in climate change, as we learned from Simon Griffiths, you remember, about a year ago, yeah? They struggle, basically. They Australians, they're adapted for arid environment, but they actually struggling and dying because of heat.
I mean, I do remember I had to like bring them inside to take the thermal images, and yeah, a lot of them were like opening their beaks or holding out their feathers, so that's a very valid point, yeah. Yes?
I'm just wondering about the models, that you have quite a big variation of the optimal strategies. Why are they? So for instance, I guess you repeated the run several times, and if the model is really able to find an optimal solution, you would accept just one point or a very small range around the so-called two optimals. But you have quite a big range for the solutions, for instance, clutch size, and in other cases, the laying. Why is that?
Yes. So each of the dots are the result of one simulation run. So when I compare how the mean changes, obviously the more simulations you do, the more it asymptotes around a value. In terms of the range of clutch sizes or the range of lay dates you're asking? Or both? Both. Yeah, so I don't know, I think it's also the fact that it's simulations, not really like an optimization. So I'm just identifying, it's sort of a random process, and I'm identifying what combination of lay date and clutch size resulted in the highest fitness, where fitness is defined as the number of offspring that fledged times by the number of caterpillars that they ate while they were in the nest.
This is just a kind of hill climbing type of optimization, that you have random parameter values, pairs of parameters for clutch size and lay date, and then just calculate that how much is the fitness, or how was the optimization done?
Yeah, so it wasn't like that. So the way it was done is we defined a uniform distribution of clutch sizes, so between 1 and 16, same sample size for each clutch size. And we had 375 as a population, a population of starting birds. For each simulation run, it was the same number, because it was just 1 year that we ran each time. So it wasn't like the years influencing each other, it was always individual years. And then the birds kind of do their things in terms of what happens with the temperature. So the temperature is linked to, for example, laying in terms of probability values. So they can sort of like choose, if there's high probability of me laying at this given temperature, then I will choose that or not. So laying date wasn't like encoded, but it kind of emerged as they were doing their things. But yeah, clutch sizes were just put in as a uniform distribution, so that's how. And then the same with how many caterpillars the chicks were eating. It was as a result of the foraging intensity of the parents, which was again probabilistic. So the actual numbers of caterpillars is like a very unique number for each nest. So that's why we always really got one solution emerging as the best, because it was a very unique number.
In this case, the laying date was not optimized. It was just hard-coded in the model. Because it was a probabilistic one,
Yeah.
it was not optimized. Right?
Yeah, no, so it's, yeah, as you say, it was probabilistic. And in that way, there was some flexibility around it, but that's why you see, yeah, a very narrow confidence interval around the laying dates, because it's kind of a...
consequence of the structure of the model.
Yeah, yeah. Yeah.
So there are, basically this is just a kind of problem search of the plot size.
Yeah. Yeah, that's right.
I had a question actually mainly related to this, so how the simulation was done. So when you showed the average optimal clutch size, that was something around 4, which is obviously less than half of what you find in natural populations, although you said that the model was parameterized for great tits. So why is this discrepancy?
Yeah, so by what I mean by parameterized for great tits, it's relationships, for instance, between on average, how many caterpillars they need to bring to the nest in order to keep the chicks alive was encoded given estimates found in the wild, like roughly 100 caterpillars per chicks, things like that, or knowing the temperature sensitivity of the birds in terms of laying. So it was parameterized with these values, but yeah, in terms of clutch size, nothing, so that was just given to the model. And I had some points about why I think that difference might be. That in the simulations we see much lower clutch sizes emerging than in the wild. And you can think of some biological explanations, like short-lived species like gray tits tend to put more effort into reproduction early in life. So that's why we might see on average the observed population laying larger clutches than what we find optimal, whereas in our simulations we were looking at individual years, so there wasn't any continuity of having to look forward to next reproductive events. And also in the wild, larger clutches can act as reproductive insurance. So actually the birds in the wild can collect other types of food for their young, whereas in the models, the caterpillar availability set a very hard limit. So once it was out, the parents had to abandon the brood. So it's kind of partly methodological as well, and leads to the next point that we had a uniform distribution of clutch sizes because we didn't want to bias the model in that sense. But it also means that a larger proportion of the population has to deal with a harder to care for clutch, rather than in the wild where we see a normal distribution. And yeah, I think I mentioned the caterpillars as well. So yeah, just some explanations, but yeah, I think it shows also your point that some of the things are a result of just hard coding it into the model rather than any like optimal solution. And yeah.
Just one related question. So when you put the points in the figures, that was the mean of one simulation? The mean of all birds in one simulation? Or
no, it was...
That was the best birds?
Yeah, the best bird, yeah. Yeah.
Thank you. Sorry, I have to leave for some stupid administration.
Thank you for coming.
All right. Any other questions? Yeah.
Maybe a small comment and a question. We did a really similar experiment with house sparrows last winter, like the one that you did with the feeders, with the foraging challenge. And interesting and sad thing is that we didn't get past the pilot because when we did the experiment in the winter, a bunch of animals died, so we had to stop the experiment, probably because the fact that they had insulin problems. And we repeated the pilot in summer, and then we couldn't manage to have proper differences between the control and experimental groups. So might be quite an interesting question how much the effect of the external temperature is in these cases. So this was an outside aviary experiment, and we just couldn't do it after this issue. The other thing is, as I just did this R package also measures the barbule length for air density.
No, it doesn't, but yeah, I
This
haven't.
made me remember a question that I had when we were still working with feathers during my masters. That it would be really interesting to see if the barb density decreases, so the barbs are growing further and further, then the barbules have to be longer to touch. And maybe there's a golden ratio somewhere, what's the optimal solution for this kind of stuff, and it would be nice to have an automatic way to measure this.
So, I mean, I can see ways that you could adapt the package to do that, like the way you just kind of use a flood fill. So once you have the position of the barbs, you use a flood fill to fill up the whole barb, and then from that you can calculate the width and the length from that. So I think you could adapt it also to do with barbules. But yeah, my idea is to release this as a package that people can use and add to, and yes, you're very welcome to figure that part out, but I haven't yet. No.
Just thought
that would be very interesting.
Wasted a bunch of autumn and winter hours just measuring feather quality metrics by hand.
But yeah, I wanted to say that our zebra finches were also housed outside actually. But yeah, they live out in outdoor aviaries all year round. So, and I think there's no, as I also looked at it, and I think others have looked at it for a longer period of time, there's no survival difference between them as such. I might be wrong, but yeah, so we still get many birds surviving in the harsh foraging conditions as well.
Well, I was selected for this environment, so is there a selection experiment to forage by hovering only?
No, so they're not really hovering, they're just flying back and forth, I think, is what they're doing. And people have measured that the way they know that they face harsher foraging costs is that they spend a much larger amount of time flying as a result, because they need to constantly fly for food. So yeah, they just fly back and forth to eat it.
But the question is that it's supposed to have a strong selection pressure on the birds in a harsh environment. So part of what you measure might be the result of selection rather than phenotypic plasticity, right? So maybe just birds that can cope with this, they just die and then...
Yeah, so these birds have been under the treatment conditions all their lives. And then some of them are also allowed to reproduce, some are not, so there's that treatment as well. And then their offspring are divided between the different condition groups, so it's not that all the offspring of a parent living in harsh conditions also lives in harsh conditions. So I think that will limit some of the adaptation. And I don't know how long it has been going on, it's maybe 7 years, I might be wrong, so yeah, I don't know if,
I had one more question about the barbule density. Is that, if I understood it well, and it looked from the illustration that you measured the barbule density and these morphological patterns of the feathers from the flight feathers, right?
Yes.
I was wondering how consistent these metrics are across the body, and especially as you interpret it as an insulation pattern, I would say that probably the downy body feathers are more important for insulation than the flight feather, and the structural pattern of the flight feather is probably more important for flight itself rather than insulation. So how consistent is it across the body?
Yes, it's something that I've been wanting to address, but I haven't, because I was torn between wanting to address this question, but also I thought it was very important that we didn't pluck the feathers, although plucking body feathers is much less of a strain on the animals than plucking flight feathers. So that's one reason why, like, you can't really use this on a live bird trying to measure their body feathers. You would have to pluck it. So I did image some museum birds, some museum specimens, but I haven't got to analyzing it yet, so that's definitely a project that I want to look into. But yeah, there are consistent, well, looking at the flight feathers, there's consistent differences between individuals, so I would guess that it's representative of the whole body feather, of the whole plumage, but yeah, I haven't addressed it directly, so it's a good point.
All right, any more questions? I think our time is up, so thanks again, Anett, for